students. My name is Sneha Mittal. I am working as an assistant professor at Ramesh Institute of Vocational and Technical Education, Greater Noida. Today in this video lecture, I am going to discuss about the anti-malarial drugs under the category of miscellaneous agents. This is a subject of medicinal chemistry 3rd under the code of BP 601T. As in my previous lectures, I have discussed many anti-malarial drugs, but there are few drugs which comes under the miscellaneous category. So, today in this video lecture, I will try to make you all understand about these miscellaneous anti-malarial drugs. Basically, in this video lecture, we are going to focus on these 4 drugs: Pyrimethamine, Artisonate, Artemisinin, and in the last AKtU. So, we will start one by one and we will try to understand these. The first drug is, first of all we will understand about the pyrimethamine. Pyrimethamine is 2,4-diaminopyrimidine. There is a substitution of 2 amino group at respective 2 and 4 position. So, we will see that pyrimethamine is a class of anti-malarial drugs which comprising of 6-membered heterocyclic ring. It is a 6-membered ring and it contains a two heterocyclic atom at this position and this second position. So, they are the 2 heterocyclic atoms which are present in a pyrimethamine drug. Pyrimethamine was developed in the 1950s, which is responsible for the inhibition of reduction of folic acid and dihydrofolic acid to the active tetrahydrofolate coenzyme. Means these drugs inhibit or are responsible for not to convert of folic acid and dihydrofolic acid to their active part, that is tetrahydrofolate coenzyme. So, we will see one by one their mechanism of action. We will see, first of all we will understand their substitution for the maximum activity. So, we find that pyrimethamine is a highly active when there is a chlorophenyl group was attached at position 5. We found that 1, 2, 3, 4, 5 and 6. So, we see that if there is a chlorophenyl, we find, sorry मैंने गलत कर दिया numbering, just give me 1 minute. We find that if we see the maximum activity is shown when there is a addition or then there is a attachment of chlorophenyl group. Phenyl means this is a phenyl ring and phenyl ring with a chloro substitution leads to the chlorophenyl group was attached at position 2 and a electron donating group, that is C2H5 at position 6, means this is a 5th position and this is a 6th position. So, we find that the presence of the electron donating group at position 6, C2 at 6, and the presence of chlorophenyl at position 5 leads to the formation of a compound which is having the maximum activity of anti-malarial drugs. Here we will understand about the structural activity relationship. In the structure activity relationship, we understand that the different substitution either leads to the increase in the activity, decrease in the activity, or sometimes they increase or decrease the toxicity or the side effects. So, we will discuss one by one. First of all, aromatic ring. Aromatic ring means this aromatic ring is directly attached to the pyrimidine ring for the maximum activity. They are stating that if is in between these two any other atom like carbon or nitrogen is inserted, then activity is decreased. So, here we are understanding that for the maximum activity, it is very important for the direct collaboration of these two rings. If there is a if suppose there is a insertion of any atoms like the nitrogen and carbon in between these two rings, then it is saying that it leads to the decrease in activity. So, there should be a direct linkage because it is decreasing the activity. Moving ahead, we will see that if the aromatic ring is replaced by the heterocyclic ring, if this respective aromatic ring is replaced by a heterocyclic ring, then the activity is reduced. We found that there is no improvement in the activity, whereas there is the reduction in the activity. Third, we will see this aromatic ring must contain a electron withdrawing group at para position. This para position is very much important for the anti-malarial activity of this drug. So, this aromatic ring must contain a electron withdrawing group at para position. Next, presence of electron donating group at position 6 is crucial for the anti-malarial activity. Next, if these two rings, the pyridine ring and the aromatic ring, if they are substituted or if they are separated by a methyl bridge, then we found that the plasmodium, that is the anti-malarial activity is reduced, but the antibacterial activity get increased. So, here we see the example of trimethoprim, in which see if these two rings are separated by a methylene group, so there is a addition of antibacterial activity. Furthermore, we will discuss about the mechanism of action. So, pyrimethamine is also responsible for the inhibition of DHFR, that is dihydrofolic reductase. Dihydrofolate reductase is very much important for the formation of nucleic acid. We will try to understand its mechanism by watching this. This is a folic acid. So, this folic acid with the help of enzyme dihydrofolate reductase changes itself to the dihydrofolic acid, and in the next step this dihydrofolic acid changes into the tetrahydrofolic acid, and in the last step this THFA, this tetrahydrofolic acid is responsible for the synthesis of nucleic acid. So, we can understand that if DHFR gets inactivated or if this dihydrofolate reductase enzyme get destructed, so there is no formation of nucleic acid and there is no survival of parasite is possible. So, it is also used with a combination of other sulfonamide sulfonamides is having a structural similarities with the PABA. So, it competes with the PABA for the synthesis of dihydrofolic acid. So, when this pyrimethamine is used with the sulfonamide, so sulfonamide competes with the PABA, inhibits the synthesis of dihydrofolic acid, whereas the pyrimethamine is responsible for the inhibition of the enzyme which is responsible for the conversion, that is dihydrofolate reductase. So, by this graph we can understand the mechanism of action of pyrimethamine very well. Next we will start the drug artemisinin and its derivative. Artemisinin and its derivative belongs to a antimalarial class which were first isolated from the plant wormwood, that is Artemisia annua. This is the plant from which these drugs were isolated at first time. These are having the antimalarial activity due to the presence of 1,4,1,2,4 trioxane ring. This 1,2,4 trioxane ring is responsible for the antimalarial property. Moving ahead, we will see the artemisinin and its derivative are tetracycline structure which is having a trioxane ring and a lactone ring in their structure. If we discuss about their molecular weight, then they are the high molecular weight compounds. The molecular weight is 282.332 gram per mole, and if we talk about the molecular formula, we see it contains the 15 carbon, 22 hydrogen and 5 atoms of oxygen. It is active against the hypnozoids form of the plasmodium. We will discuss about the SAR, structure activity relation of the artemisinin. It is a main compound which is used for the development or which is used for the development of many products with enhanced and activity with less side effect. Means by doing the several modification, we can get few more compounds which are more potent and which are having more better anti-malarial activity with reduced side effect. So, we will see here first of all the lactone ring of artemisinin is reduced to dihydroartemisinin. First modification which we found here, we see that in artemisinin there is a presence of this C double bond O group, that is a ketone group. So, this ketone group was changed into the hydroxy group. So, this was a artemisinin and this is a formation of dihydroartemisinin. So, this lactone ring of artemisinin is reduced to the dihydroartemisinin, which is used to prepare the prodrug which is having high solubility. If we see the artemisinin, it is having a poor solubility. But this moiety is available with high solubility. So, through which we can understand that by doing the reduction of this ketone group to hydroxyl group leads to the high solubility. Next, the hydroxyl group can be alkylated to produce oil soluble ethyl derivative. Here we see this OH group. This OH group can be further substituted to form the more lipid soluble drugs. Like we see the examples of artemether and artether. Artemether and artether. These are the few drugs which are more oil soluble because there is a substitution at OH हाइड्रोक्सिल ग्रुप, जो हाइड्रोक्सिल ग्रुप का सब्स्टिट्यूशन हुआ था उससे हमें और ज़्यादा लिपिड सॉल्युबल ड्रग्स हमें प्रोड्यूस हो रही हैं. We'll see esterification of the hydroxyl group with succinic acid gives the water soluble derivatives like artesunate. So, here we will see if there was a OH. So, if we do the substitution at this position and we form the different compounds. So, we can see if this R is substituted with CH3. So, there is a production of methoxy group. So, this compound is known as the artemether, which is having more oil solubility property or which is more oil soluble ether derivative. Next, we will see if there is a substitution of this R with CH2 CH3, it leads to the formation of artether. And last, we will see that if there is a substitution with succinic acid, then there is a formation of it leads to the formation of artesunate. So, these are the few substitution which can be done at the this OR position. Next, we will discuss about the artesunate. Artesunate is also known as artesunate or artesonic acid. These are the synonyms of artesunate. Artesunate is also a artemisinin derivative. This structure is similar to the artemisinin, just it is having a slight change that it contains a 1,2-dioxypane fused to an octahydrobenzopyrene moiety. So, here we will see that a tetracyclic compound, this is a tetracyclic compound, it is having a 1, 2 and 3. The 3 cyclic rings are present in the structure, and here we will see that the structure is based on the artemisinin, a tetracyclic compound that contains 1,2-dioxypane fused to an octahydrobenzopyrene moiety. The internal peroxide bridge is believed to be responsible for the artemisinin. This bridge, endoperoxide bridge, is responsible for the antimalarial activity. If we talk about the solubility of artesunate, so we find that it is soluble in water and it contains antimalarial, antiviral and antineoplastic activity. Next, moving ahead towards the artemether, we will see that if the artemisinin, where this ketone group is present, if it is get reduced and gets methylated. So, this first of all there is a formation of OH, that is अगर हम इसका reduction करते हैं, so ketone group gets converted into the hydroxyl group, and then after that there is a removal of hydrogen and addition of methyl group. This is known as the methylation. So, after methylation there is a formation of artemether. It is same to the artemisinin, it is almost same to the artemisinin, just it is having a slight difference that it contains this methyl group. This methyl group is responsible for its higher lipid solubility property. It makes this methyl groups makes these drugs more this methyl group makes the drugs more oil soluble. Next we will see to increase its action, it is given in combination with lumefantrine. अगर इसके एक्शन को हमें बढ़ाना है तो ये सिनर्जिस्टिक एक्शन दिखाता है with the lumefantrine. It is rapidly and extensively metabolized in the liver to dihydroartemisinin. The same as I discussed before, it the ketone group gets converted into the hydroxyl group. An active agent which appears to be responsible for most of the antibacterial activity. Next आर्टिमिथर में हम आगे डिस्कस करते हैं. जैसे हम आर्टिमिथर को हम पहले भी हम डिस्कस कर चुके हैं कि अगर हम इसका रिडक्शन कर दें या फिर हम इसको मिथाइलेटेड कर दें, सो देयर इज़ अ फॉर्मेशन ऑफ आर्टिमिथर विच इज़ अ मोर lipid soluble drug. Now we'll discuss the mechanism of action. As I discussed that there is a formation of 1, 2, 3 and 4. So there is a formation of 1, 2, 4 trioxane bridge. There is a presence of 1, 2, 4 trioxane bridge. So this bridge is known as endoperoxide bridge, which is responsible for the antimalarial activity. Now we will discuss how it is responsible. It interact with the heme part in the parasite. We know the hemoglobin gets distributed into two parts, heme and globin. Globin is used by the plasmodium as a nutritional value, whereas heme part is dangerous to the parasite. So this heme iron cleaves this endoperoxide bridge. इस bridge को वो तोड़ने की कोशिश करता है and there is a formation of highly reactive free radicals. These free radicals damage the parasite's membrane through the covalent bonding. वो उसके साथ covalent bonding कर लेते हैं and there is a death, there is the breakdown of the membrane of parasites through this highly reactive free radicals. So through this mechanism, this bacteria, this plasmodium gets destructed and ultimately the death takes place. If we talk about the metabolism, so artesunate is metabolized to the active DHA in the liver. The endoperoxide bridge is responsible for the antimalarial activity because it destructs the membrane by binding them covalently. Reactions with these free radicals can also lead to alkylation of parasitic proteins. Next uses, it is used for the moderate malaria. It is recommended by the WHO for the first line treatment of malaria. It is also recommended in the form of injection having a concentration 2.4 mg per kg administered. The only side effect which was found is the renal impairment. The next drug we discuss the atovaquone. एटोवाक्यून का अगर हम मॉलिकुलर फॉर्मूले की बात करते हैं C22H19ClO3, it is very much similar to the previous drugs, but it is having a hydroxynapthaquinone. This is a napthaquinone ring in which it is having a hydroxy substitution. It is used for the prevention and treatment of pneumonia, that is the pneumonocytis jirovecii. So it is used as the prevention as well as for the treatment of Plasmodium falciparum malaria. If we talk about the mechanism, so in the electron transport chain there are the many complex 1, 2, 3 and 4. So this drug very much similar to this coenzyme and inhibit the electron transport chain. Why? Because This drug is structurally similar to the inner mitochondrial protein, that is ubiquinone. So, this ubiquinone is just present adjacent the complex 2 and hence it inhibits the electron transport chain by binding to the complex 3rd, and it inhibits the electron transport chain at this stage only. It is used in the combination of proguanil in the ratio of 2.5:1. It prevents the DNA synthesis, leads to the death of parasites. Furthermore, we will discuss about the uses. So, we will see that it is used against the falciparum through the inhibition of electron transport chain at the level of cytochrome BC1 complex. As I discussed earlier, there are the 4 complex 1, 2, 3 and 4. So, it interfere with the electron transport chain. Cause collapse of the parasite mitochondrial membrane potential, and it is also used for the treatment and prophylaxis of malaria. So, by this we can understand that it is helpful in treating the many antimalarial, it is used as a antimalarial drugs. Side effects we see nausea, vomiting, diarrhea, headache, dizziness are the few side effects which are seen with these drugs. Thank you students. Have a nice day.